students. Yesterday we began our notes on genetic engineering, and today we finish our notes on genetic engineering. And yesterday we went over two techniques that genetic engineering can be accomplished through. And today we are going to pick up where we left off yesterday with number 3, and that is gene targeting. I find this the most fascinating form or technique of genetic engineering. I think this is really interesting as we discuss it here now. Gene targeting is the isolation of a particular gene to either turn it on or turn it off. Now, what exactly does this mean? Well, first, let me give you an example. As you may know, I have identical twin daughters. And so, as identical twins, they have the same DNA. One of them could commit a crime and blame it on the other one, and no one would be the wiser. Not that I would ever encourage my daughters to commit any crimes, but they both have the same DNA, although their fingerprints are different. But here's the deal. My daughters, even though they're identical twins, are completely different people. And they have grown and developed differently over their lifetimes. They are not the same person. They are different people, even though they share the same DNA. And part of the reason why they're different has to do with the fact that their DNA, sections of their DNA, certain genes, have been triggered on and or off at different times in their life. And I just find it as a scientist kind of fascinating to think about, well, why have some of their genes turned on or off at different times if they have the same DNA and they grew up in the same house and they eat the same food, et cetera, but somehow, for some reason, these genes have been targeted to be turned on and turned off at different times. I just find that fascinating. Now, in the sense of, in the larger context of gene targeting, here we might have a particular gene, and I'm not sure why this isn't showing up on the screen, but here we go. So we might have a particular gene here that is the signal for something. Gene targeting identifies this particular gene and either turns it on or turns it off, and either activates or deactivates it, you might say. And in so doing, if scientists are able to do this more effectively, and they're learning every day, they could actually turn off the gene that controls a certain disease, perhaps, maybe even cancer itself. To be able to target a very specific gene, if that gene can be identified and then isolated and then turned off, imagine the possibilities of saving people's lives. But then by the same token, you get into the ethical dilemma of, well, if you could save people's lives by turning off certain genes, are there certain genes that could be turned on in order to maximize someone's life or give them superiority over other people whose genes are not turned on? It's a big ethical debate in the scientific and the medical fields as to what to do or what not to do. So that is gene targeting. So that brings us to number 4. Number 4 is called transformation. Transformation is is the process of injecting foreign DNA into a cell in order to allow the cell's DNA to respond to the foreign DNA in a desirable way. This seems to be a little bit more complicated, but it's almost in a way similar to how vaccinations work. You've all been vaccinated against something, whether from childhood or at childhood to later in your life. But a vaccination is intended to inject a foreign substance into your body so that your body builds antibodies against whatever that is, so that your body builds up immunity to it. I mean, that's how a vaccination works. Well, with transformation, it is a specific foreign DNA segment that is injected into the cell of a healthy individual. And we're not just talking about humans, we could be talking about bacteria or bugs or whatever the critter might be, in order to allow the cell's DNA to respond to the foreign DNA in a desirable way. In other words, it's essentially turning bacteria, when it's used in bacteria, into DNA factories. So let's use the analogy or the example of an antibiotic or an antivirus, a, an injection of some kind that we might desire to protect us against some dangerous strain of a virus or a bacterial infection. Well, instead of using human guinea pigs, we use bacterial factories. And we can take the interested DNA, inject it into bacteria, let the bacteria produce the serum or the antivenom or whatever the substance might be that could later be injected into people in order to achieve the desired result. So we're basically using bacteria to achieve a desired result. There's a famous experiment called the Griffiths experiment, which does just this. It takes a segment of DNA and injects it into healthy cells of a mouse, which are then placed in a petri dish in order to grow. And over the course of the experiment, it is the bacteria that are producing the desired effect. Again, we'll see examples of these in the video segments coming when we are finished with our notes. And speaking of our notes, that brings us to number 5. Number 5 is called regeneration. Now, this is pretty outrageous when you think about it, but regeneration in the context of genetic engineering, not in the context of like starfish or sponges have the ability to regenerate, but genetic engineering regeneration is when there is an injection of a desirable DNA into a stem cell, and the stem cell then goes on to produce an organ or a complete organism with desired traits. Now, check out the picture here. The picture on the left is simply a petri dish where things are being injected. Desirable DNA is being injected into the arm of a mouse. But look at the right hand side here. This is actually being done. Here we have a mouse growing a human ear, growing a body part. Why? Well, as I recall reading in this particular study, there was a burn victim that had their ears just burned off their head. And in order to look more normal, they're utilizing this regeneration technology to take DNA from the patient's body in order to grow an ear on a mouse in order to provide the circulatory system for the ear to grow to full size. But the ear is being grown with the DNA of the donor, with the patient, so that when attached, the hope is that the recipient, who is also the donor of the DNA, their body doesn't reject the organ or in this case, the body part of an ear, so that they can live the rest of their life without having to take organ rejection medication so that their body doesn't attack it as a foreign substance, which is the case today. Some of you may even know someone who has had an organ transplant, and there is a donor and a donee. The recipient or the donee gets an organ donated from someone else, but for the rest of their life, they have to take anti rejection medication because their body, even though they might have received an organ like a heart to save their life, the rest of their body views that as a foreign invader and tries to kill that heart that is now keeping them alive. And as a result, they have to take medication daily for the rest of their life to inhibit their own immune system from attacking the organ that's keeping them alive. But with this particular technology, if it can be utilized to grow lost body parts, including organs of someone with their own DNA, then their own body won't reject the regenerated organ from their own body, even though it might be grown externally in a lab, in a petri dish, or on a donor animal like a mouse. Once it's placed into the recipient's body, if it's made up with their own DNA in the first place, their body won't reject it. That's pretty fascinating when you think about it. In fact, they're suggesting now, and when I say they, I mean the futuristic scientists among us, are suggesting that in the future, if you lose a limb or an organ, you might fairly easily be able to provide a donation of your DNA, and they'll be able to 3D print a replacement body part or organ using your DNA. And when it's attached or inserted to your body won't reject it because it is your own DNA. That to me, that's just mind boggling. But perhaps by the time you're my age, that'll seem like second nature. You'll be able to go to your local Walmart store. If you had an accident and you cut your finger off, oh, I lost a finger. Let me just go down to Walmart and print myself a new one. Who knows? Maybe it'll be mail ordered and delivered to you by a drone and automatically reattached. Who knows what the future holds? We do know one thing that the present holds, and that is a debate about genetically modified organisms. There is a debate raging across our country right now because we are living in the forefront, the early days of genetic engineering. And so there are a number of people that are concerned about what we refer to as GMOs, or genetically modified organisms. And generally speaking, these are organisms with intentionally modified organisms with intentionally modified or altered DNA for our use. Now, if this scares you, don't let it scare you too much, because you're already uh, a guinea pig in this regard. You have been ingesting genetically modified plants and food products from genetically modified plants your entire life. There's no question that you have been a guinea pig in this regard. We have been experimenting with genetically modified organisms, with microbes, plants, and animals for a number of years now. The real debate, uh, the bigger debate currently, is what is the impact on human beings and our DNA as a result of these GMOs that we are ingesting, because they just haven't been around very long for us to do long-term studies on them. So, there are a number of genetically modified foods that you probably are consuming on a regular basis, especially in our country, because we're one of only a couple countries in the world that doesn't require, at least at present, genetically modified organism labeling. Many of our tomatoes rice, sweet corn, wheat, cotton, sugar beets, summer squash, etc. A lot of these things are already genetically modified. And you can go to a website here, olmag.co/GMO foods. Now, this is, I believe, a United Kingdom website, if I recall correctly. But you can go online and Google genetically modified foods, GMOs, and find out more about this controversy, which includes some major points. Number one, at least in this country, there's a controversy right now about labeling the foods. As of the current date, we don't have any federal or national guidelines requiring GMOs to be labeled GMOs. And that concerns some people because some folks want to know what they are consuming. Many other nations in the world require GMO labeling. So it's probably only a matter of time before our food in the States will be required to be labeled genetically modified as well. There's concern against consuming them, whether they are safe to be consumed, whether they will have any impact on our own DNA or not. We simply don't know the long term effects because there hasn't been long term testing, because the technology itself right now is admittedly very young. And so we have a long way to go before we know really what any long term effects might be with GMOs. So here's a quick little map showing some of the major countries in the world. And you can see that the US and Canada here are currently two countries that do not require labeling. Many other major countries do require labeling. And as I said, it's probably just a matter of time, maybe even by the time you see this video, I don't know, we'll have required GMO labeling as well. Now, thankfully, there is some time remaining after this video, so you'll have opportunity to watch a couple more videos and debate this issue of GMOs before your lab activity tomorrow. So until then, I'll say bye-bye.